Right. Thank you so much. And can you hear me all right and see slides all right coming through okay?
Sounds great.
Wonderful. Great. So really good evening in the UK, good afternoon for any of those here out in the US, and then of course a little bit still good morning out on the west coast of the US. It's a pleasure. It's an honor to be at this institute where many great speakers over the past several hundred years have announced really big ideas, big developments in science, and extraordinary discovery. So I'm humbled to be in the same stage as many of the great scientists over the years, and we'll walk you through some of the work that we've done in our laboratory in the past few years, and then also a vision, as the name implies, over the next several hundred years of what we're planning and we're building towards, and that I think is really a hope and a dream for humanity's future, to which we will all, I think, contribute and hopefully benefit from. So I'll start with a bit of background. Is just some figures from the book, again, that you've heard is just out today, is, for example, this one where we know there's basically an exponential increase in the number of objects launched, which includes spacecraft and satellites, into the atmosphere, now orbiting the planet. So this is, over the years, you can see for a little while it was the US and mostly Russia. It's fluctuated. There's a bit more of other countries now coming in. But now this is an exponential increase, even super exponential. So now we have objects being launched not just around Earth orbit, but also, as you all have probably seen, just recently we also had the little small helicopter that was in the Perseverance rover came out of its belly, went out into the Martian surface, and then did a little flight up, did a little turn, took a look around, showed that it was able to sustain flight just fine, and then eventually also decided to come back down and land then also on the Martian surface, really as one of the first and most exciting examples of powered flight on a different planet. So really went out without a hitch. And it really means that we are now entering this really exciting phase of where we can imagine not just going to Mars temporarily, but even maybe going there permanently. And so this is an image of NASA's goals to actually get to Mars. And again, not just to visit, but to stay permanently and become a multiplanetary species, and to really explore. And really, I like to say this is not plan B, this is really plan A. This is something that is really native to humans in terms of their desire for exploration. And what I'll argue to you later today in this lecture is that this is also a moral duty we have to go to other planets, and I'll get into that in a little bit. But first, I want to start with some of the other science that's really extraordinary and driving us to an accelerated and extraordinary phase of science. And one of these is also just the fact that if you think of exploring space, that's going really fast, even just exploring the DNA in our cells, inside of each one of our cells, is now faster than ever before. This represents the chart of the cost of DNA sequencing over the past 20 years. You can see it used to be hundreds of millions of dollars to sequence a human genome. It's now less than 1,000, getting closer and closer to $100 a genome. And this is moving even faster than Moore's law, which has led to rapid acceleration of the density and speed of transistors on microchips. And it's why your smartphone that you might have on your pocket right now is faster than supercomputers of the 70s and 80s. So it's really extraordinary speed of technological innovation, the fastest that's ever occurred in human history. And this has let us really begin to discover all sorts of new things. So for example, even just saying how many genes are in the human genome, original numbers used to be 20,000, maybe 30,000. We now have actually almost 60,000 human genes that have been discovered, and 10,000 of those are just in the last 10 years. So we're discovering new genes to this day. The fundamental components of the human genome are still being cataloged and discovered. Also, you can think about how many species are we finding with these new sequencing methods. And here we're getting, tens of hundreds or hundreds of thousands of new species coming out per year. And it's expected that this will continue really out into the 2030s and 2050s, just because there's so much left we're still finding. And so you can see birds, we're probably not going to find any more birds, but they're kind of big and easy to spot. But a lot of the other smaller creatures on this planet, we're going to continue to find at a really rapid pace, driven a lot by these sequencing type methods. And not just on Earth, we're even finding some in space. We just published a couple of weeks ago some interesting bacteria that are on the space station that could even look like survive in the high radiation environment of space. And so what this means is that every day there's more sequence data. Every day we're discovering new species. There's a greater capacity every given day to discover new things. That actually means that every single day that I wake up as a geneticist is actually the best day to be a geneticist. And that's actually true every single day that I wake up, because there's more data. There's the most data on that day. So there's the greatest chance for discovery, for rapid annotation, for using the genetic catalog of all life to better help life survive or adapt to different environments. And so that's kind of, as the intro, I'll say what I put into the beginning of this book as well, which is my really a dream of hope for humanity, as well as a proposed duty for humanity. I think something that we can all share. And so this is, again, just came out today. So really excited to be here today to talk about it. But I broke this out into 10 phases. And I actually first wrote this about 10 years ago on the lab's webpage. I said we should break this into 10 phases of what I think will happen in the next 500 years. It's also the structure of the book. And I want to walk you through the first phase, which is most of what's happened in the past 10 years, which is, as I just alluded to, more annotation of the human genome, finding the actual genes and the regions of the genome that we shouldn't disturb. We're going to begin outlier studies of humans in the early stages of synthetic and systems biology. And what I mean by that is, is not just in the U.S. context, but it's become a global quest. So now I'm going to walk you through some of the work we've published and done that deal with twin studies and some of our astronaut missions. And then I'll walk you through the next 480 or 90 years or so. But first, it's worth pointing out, it's not just NASA. China and India is racing to get to the moon. You can see even the Israelis had a lander that got onto the moon. It actually spilled some tardigrades onto the surface of the moon. These things are called, water bears, these tardigrades that can actually survive in the vacuum of space, these cool little creatures. Some people think they're creepy. Some people think they're gross. I think they're quite beautiful, still a little bit gross, but quite intriguing. And you can see them here, but they're amazing. They survive in the vacuum of space, high levels of radiation. I'll come back to them in a little bit. But we've even seen recently the Chinese mission, the Tiangong-4, put plants on the moon, and cotton plants sprouted on the moon. We've seen that the European Space Agency has begun to give grants for lunar architects to build moon bases. This is a rendering from an architectural firm that has been awarded funds to literally build a base on the moon. So if you're thinking of your children and you're meeting kids, later today or this coming week and weekend, and a child says to you, well, I want to be a lunar architect when I grow up, you can say that is a legitimate job. You should plan for that because it's an actual job, as you can see here. And so there's a lot of excitement to build things on the moon, to get to the moon, to go to Mars. But we also know from some of our work and many other prior NASA missions that there is some risk. There is a biological risk to go to the moon and hang out there, like you can see here. And this was first discovered by some of the Apollo missions. Particularly Neil Armstrong wore these, what are called these nuclear tracing packs on his ankles, which you can see here. These are high energy particles zipping through these packs, these foils, which also means they were zipping through his cells and causing DNA damage. So we know that this is one of the hazards of spaceflight, is radiation. And we also want to put this into the context of other changes that just occur as a consequence of time. So for example, we know that telomeres shrink as you age. You can see these sort of caps at the end of your chromosomes, which contain all your DNA. Over time, your telomeres shorten, and then eventually the cells stop dividing. So we know that this is a normal part of drift of the biology, where your telomeres get shorter. And we also want to look at, DNA, RNA, and protein in astronauts, and particularly also the regulatory layer behind DNA, which is the epigenome, and see how DNA is packaged and controlled and regulated. And the other thing we can look at that is when is also DNA damaged. So we can see the genome is ACGT, the genetic code of life, is, you can see here in black, but the epigenome are these small chemical tweaks here in red that can control when and how and where DNA is used, or in the case of adeoxyguanosine, show when it's been damaged or disrupted. And so it's really, we can start to measure all these things with exquisite detail today with new methods. But some of the discoveries we've known for years about, say, 5-methylcytosine, this epigenetic mark. We know, for example, that epigenetic twins are more different as they age. But epigenetically, you can see the number of epigenetic differences for 50-year-old identical twins versus 3-year-old identical twins. And this is true that, genetically they accumulate mutations, but epigenetically you can say they become more different over time. And then, we also can see that, mutations that occur is something called clonal hematopoiesis, that in your bones, actually, where the blood is made, hematopoiesis, you have these clones, basically cells that are carrying mutations and expanding over time. So that eventually we almost all probably carry some degree of these mutations in our bones and in our cells, because we've just gotten, essentially we've lived on Earth long enough, you'll get mutated. We're all a little bit mutants. But this has been known for a while, and it can even predict in some cases, maybe a decade before you get leukemia, that you're on that track. So we're using this data now to look at the trajectory of mutations, not just the presence or absence of mutations. And so this is kind of what we know what normally happens to humans on Earth. But what about in space? So in particular, we had a unique opportunity to answer this question with identical twins In this case, it's Mark and Scott Kelly, and you can see here Scott on the right and Mark on the left. Mark always says he's the handsome one with the mustache, but otherwise they're identical. And also, Mark Kelly recently became a senator, so now we have a senator genome project in the lab. But when we launched this study, this was a unique opportunity to design a special patch for NASA. They were very excited to put a patch that had epigenetic marks in the middle, you can see, and identical twins. And we were one of the 10 teams selected for this mission, as well as some other missions I'll tell you about today. But we, launched Scott out into space in 2015. So he made his way up to the space station for a year. And during that mission, we had all kinds of experiments we wanted to do. Some of them included, for example, we wanted to see what does the immune system do in space. For example, if you give yourself a vaccine, in this case, you can see here Scott making the vaccine face, I would say, and you're giving yourself a multivalent flu vaccine. And then you can see here once finishing the injection, then looking up, getting it recorded, a little bit of an ouch there, but he did get it in, successfully gave himself a vaccine shot. And we also could measure the immune system response afterward and kind of successfully gave himself that shot, gave everybody kind of a good thumbs up and said that things are going okay. So it was great. We did that experiment. We did, to look at his blood, we did frozen blood that was spun on the space station, but we also got fresh blood draws. Here best demonstrated by astronaut Koichi Wakata, who shows when you draw blood in zero gravity, it's a little bit different than what it is on Earth. You can see the tube kind of moves around. The butterfly needle still sticks in the vein, but you have positive pressure inside your body. You have negative pressure inside the tube. So it's very easy to actually do the blood draw. Granted, he's doing it on an empty stomach and in zero gravity by himself, but no problem. He's an astronaut. And you can see not just one tube, but we have sometimes get 8 or 10 tubes. We got some of these fresh blood tubes dropped in, launched back through the atmosphere, were deployed. Some of them would land in Kazakhstan and retro thrusters would fire. We then get them brought back, repatriated by helicopter and brought back to NASA. And we got cells that only 36 hours prior had been in orbit. So very easy to get very fresh blood from space, as long as you have an entire space agency working with you. So this was a really extraordinary effort and great work by NASA and the ops team to get this done. So it let us look at live cells very readily. So after a year in the mission, though, eventually Scott had to come home. You can see here some of the images when he's undocking from the space station after 1 year. This is the Russian telemetry data. You can see the space down below. You can see the Earth right there, kind of passing by below space. And eventually he undocked and the Soyuz capsule made its way through the atmosphere. The chute deployed, and we're very excited that he made his way safely back to Kazakhstan at the time because we're using the Russian rockets to get to and from the space station. Made it back. We're very glad. He kind of gave us another thumbs up when he got back. But what's interesting is when he did get back, he had a really unique response. He said that his skin felt like it was on fire, and he basically had rashes and discoloration anywhere they had contact. He basically had to walk around, almost nude for a while because he just couldn't touch anything. Anywhere he would sit, it would break out into a rash. He felt like he wanted to go to the emergency room. It felt really awful. Even just the weight of clothing on his skin was enough to break out into a rash because his body hadn't felt the weight of a shirt in a year. So basically, we could see this inflammation, we could see these changes, but then we wondered, why? At a molecular level, what was happening to him, and could we learn lessons from this mission to apply to future missions? So in particular, we know that some things did change, like he got 2 inches taller because of the lack of gravitational force, and 2 liters of fluid moved into his upper body. So we know that occurred. But at the molecular level, we wanted to really detail what happens. We want to really do a multiomics analysis of human spaceflight. And what I mean by that is we're using all these different tools of modern molecular biology to rigorously and deeply understand what happens to the body in space. So in particular, if we're looking at someone on Earth or someone in the space station, for you want to look across cells, chromosomes, telomeres, you can see the DNA, look at all these features. And the way we did that is we did genetic testing, we did epigenetic testing, we did RNA mapping, we did protein, chromatin, antibody, B cell, T cells, telomeres, cytokines, metabolomics, microbiome, cognition, and vasculature measures all over the course, essentially, of 2.5 years, about every few weeks, to really get a longitudinal profile of what happened inside of his body. And what we found, and we just published some of this work a year and a half ago in Science, was this is across all these omes. So this is the microbiome, the metabolome, proteome, the transcriptome, and the genome, all these omics measures. We could actually see that some metrics increased in unison with each other. Other clusters went up or down over the course of the mission. But we could see some things were surprising, like we could see spikes in some of the cognition that were changing in flight. Some things actually got better in some cases, others got worse. But the really striking thing was his telomeres actually got longer in space, whereas we hypothesized they would get shorter. And this work with Susan Bailey, where she led the effort on telomeres, we thought, well, that was kind of opposite. So we did multiple site validation, did characterization, worked with her, and also did several follow-up studies. The first of which was just to see, can we confirm this in 2 other astronauts? And indeed we could. And then we also worked to say, well, what if we actually did this to see, like, what is this the same as like climbing a mountain? Like if you climb Mount Everest, is that the same thing as going to the space station in terms of the biology and also in terms of the telomeres? And here we worked with Matt Moniz, and he actually had a twin who stayed at sea level, and he had another friend who had a twin who also stayed at sea level. So we did a different twin study on Mount Everest. In here too, in this environment of low dose radiation, we think about also essentially the hypoxia that might be there and the stress on the body. They too got longer telomeres, you can see here. So we did this also on 11 other astronauts, and we've consistently seen that telomere lengthening, it's this change of the genome, is something that's a consequence of spaceflight, or at least associated with the changes during spaceflight. And so we wanted to look a little bit more at this and try and figure out what was happening. So we looked at all the genes that went up or went down, and you can see here in purple and orange, these are genes that are increasing or decreasing their activity. And the ones that changed in flight, we could see were those involving DNA repair or the immune system, but some really surprising genes involved hypercapnia, which essentially, we could see, there have been some reports about carbon dioxide on the space station. And Scott Kelly complained about it a bit in his book, is that sometimes when the CO2 levels, if the scrubbers would break down, he would get these headaches. And so we checked with NASA and the flight logs. Most of the carbon dioxide and oxygen levels were pretty stable during the mission. So we didn't think that that was necessarily the cause, but we talked with other astronauts and asked them, do you ever experience this? And they don't necessarily experience it normally, but when they do, it's sometimes just because they're not next to a fan to keep the air circulating in zero gravity. Because on the space station, air does not move the same way it does on Earth. And so if you blow bubbles in space, it's not like what happens again on Earth. They can actually stay sometimes right in front of your face, creating these interesting clouds of mini carbon dioxide, cumulus clouds right by your face. And that's with some of the hypoxia we think we measured inside of his blood work. And so it was a really, intriguing side effect of just breathing air in space is different. And so looking back again at his body, we want to see, well, what happened when he got back to Earth? So when he did return to Earth, we could see all these different markers of what are called cytokines. These are the body's inflammation markers in response that you'll see often in response to infection or stress on the body. For example, interleukin 6 down here is something you'll see spiked up in COVID patients, but it will spike up and stay up for days and days or even weeks, or C-reactive protein you'll see spike up. And so we could actually see some of these markers of inflammation. Some of them persisted during the mission. Others really spiked up only at the day of landing. And so really intriguing differences for all these cytokine markers. Really his body struggling to say, okay, I have pro-inflammatory and anti-inflammatory markers you can see here. But in general, they're all pushing towards muscles getting regenerated and muscles coming back into use. So we can see this is probably what explained why his ankles swelled up to the size of basketballs. We think is because of likely these markers that we saw in flight. Okay, so a few other things we want to look at on the mission is, okay, the epigenetic age. I mentioned that twins epigenetically age more distinctly as they get older, but we could see here his epigenetic age, Scott here in blue, Mark in green, that for the most part, they were pretty stable actually. So we don't think his epigenetic changed that much in the mission. Certainly didn't get too much older, which is good news. But we also want to look at any mutations in his DNA. Did we see changes in essentially the presence of any mutation? Then we looked at here is this sort of what's called the allele frequency, or really this represents the numbers of cells that carry the mutation in the blood. And we could see here that this TET2 mutant actually, the mutation, the amount of it went down in flight. You see this gray part, and then kind of stayed down. So we thought, well, that's kind of interesting. In some ways, he actually got, if you think about it, he got taller and he got a little bit younger in space in some regards. But the height went down when he got back to Earth, and also when we looked longitudinally, we could see that this mutation in TET2, for example, looks like it came back up. So we could actually see that, it's just like a mole on your back. It's not a question of do you have a mole? It's more, is that getting bigger? Is it changing? Is it at all looking worse? And we're looking at the same thing here for monitoring Scott and Mark and other astronauts long term. Is we want to just keep an eye essentially on all aspects of their genome and their other omics processes. And so across all those genomes, all the epigenomes, the transcriptome, metabolome, microbiome, all these omics you can see here, for the most part, they actually recovered, and is generally good news that it seems to be the body is extraordinarily adaptive and has really rapid plasticity for a lot of the stress of spaceflight. So a bit of good news. So that's kind of what we've done. That's what we've been up to for the last, I'd say, 10 years and several missions we've done. But there's a few other ones I want to highlight that are related that will really help us think about the next phase, or phase 2, which is start to look at more astronaut studies, look at new integration of elements into mammalian genomes, and eventually get boots on Mars. So along the lines, one of the first things we're going to be doing is getting more samples. There's only 583 humans who have ever been to space. So we have a limit in how big our study can be, but we're rapidly expanding those that are joining. So for example, we're working with SpaceX on their Inspiration 4 mission. You can see here sort of their latest website. And so that flight will be in September of this year. So getting more samples from SpaceX to repeat some of these same measures. We're also working again with NASA. One of our missions was selected to look at clonal hematopoiesis and other omics measures for astronauts. So that is ongoing as well. And the other thing we're working on is, thinking, can we do these diagnostics faster, right? Instead of having to bring everything back to Earth, could we do just, sequencing in space? So we started to plan this mission out with Aaron Burton and Sarah Wallace at NASA and said, okay, well, if we have a mission called the Biomolecule Sequencer mission to sequence DNA in space, a space-based DNA sequencer, we're probably going to need a patch. And said, okay, great, we got this patch set up. So you can see here's a little Oxford nanopore. It's actually built in the UK. So fantastic little nanopore sequencer that we got. Said, okay, can we get this up into space and use it to sequence DNA? Work with Oxford nanopore. And as we were planning out the mission, setting up some resupplies for the twins study, one of the rockets that was carrying it actually exploded on the way to the space station, which is very unfortunate. No one died in this explosion, but all the supplies and reagents were lost. And I got a really nice letter the following week from NASA that said, Dr. Mason, Scott said space is hard and we're sorry that we blew up your supplies, but hang in there, keep the faith, and we will get the supplies back up there. I was very touched by this letter. It's like they really, if you get if a grant gets canceled by some other agencies, they don't even send you a nice letter. They just say, sorry, you have no more money. We're going to take away your money. So this was very, very flattering and sort of heartwarming from NASA. And then, we thought, well, in the meantime, let's see if we can get it working instead of in space. What if we did the parabolic flight simulator, which is this, vomit comet, they call it, or the plane that's been emptied out, and you get little bursts of microgravity, about 10 to 15 seconds. And we worked with Andy Feinberg, who's a friend and collaborator, to say, well, let's try and do pipetting in the parabolic flight simulator and see if we can actually get pipetting to work in zero gravity. So you can see here, normally tips are supposed to stay there inside this little box. They did not stay there. He's trying to, is currently in zero gravity, trying to do the pipetting, moving the fluid around, showing that it's possible, keeping his focus, and the tubes are flying around. Eventually you get to the bottom of the parabolic flight, and then all the tubes crash. He kept his composure, kept his good spirits. We managed to load the min-ion in zero gravity, showed that it was possible to sequence in zero gravity for the first time. And then right after that, we worked with the BC mission planners to get Kate Rubens to get training to load the flow cell and do sequencing in space. And she then safely made her way up to the space station. And we're very anxious. Everything went fine on the follow-up missions. And then actually she got up there, showed it was possible to sequence in space very readily. So now can now this be as flight validated hardware, which we're now continuously using to sequence in space, actually. It's very exciting progress. And this means that we can sequence there, but we've all started to think about, well, what if we, could think about sequencing elsewhere? So for example, in the clean room, what if we could bring a sequencer and send something to Mars like a helicopter, and we could start to look at what other DNA might be there. And so you might be asking, well, what DNA could be on a rover that we just sent to Mars? Here's the Perseverance rover before it went to Mars. You can see here the engineers working. This guy's listening to some music, perhaps, we're not sure. But you can see, they're excited. They're engineers. They might get a little excited and, maybe they'll take a picture with the robot that they're going to send to Mars because, they're people. And if you think about that, what that means is when the rover essentially is there over the next few years, it's going to actually collect some samples, package them up. It's going to wander around some of these canyons, look around the landscape. There's some beautiful pictures of Mars. This is another picture. This looks like it actually could be from Earth, but this is actually from Mars. And the rover is going to roam around and look in the horizon, try and find a spot to grab some samples, package them up, dig in the regolith. Here you can see an actual picture from the soil, and then look back in that evening at Earth, where eventually it's going to have the samples go back to. This is what the Earth looks like from Mars. And the Mars sample return mission should, from the European Space Agency, bring samples back to Earth in about the year 2032. And when it does, we have a question of, well, is there new life? Is there life on Mars? Do we see any DNA, or do we see DNA of anyone who was working on the robot? So to do that, we're basically building an index of all the DNA that we've ever sequenced from this clean room and from this rover, and basically building a genetic map of anything that's ever been found on Earth. And so this is something we call planetary genomics, because it is the genetic map of anything ever found on the index of Earth. And so this is what's part of called planetary protection, is to see, do we have any forward contamination to other planets or backwards contamination to Earth? And so we've just recently worked with NASA to get essentially a framework to map any organism that might be able to survive the trip to and from Mars, and then also built the computational infrastructure. So basically any piece of DNA that's ever been observed on Earth with any of our studies or any other published study, we can now create an index where we map it and look at it very quickly. So for example, just to show you what it would look like here, you can see essentially in some of my own slides, I'll pull up our package here, is, this camera index, what it looks like is essentially anytime you want to type in, we're not doing our studio now, but if you pull up the graph, you can say, I would like to look and see, did I have any of these databases? Anytime I saw this with Gunnar Rauch and also with Andre Callas and Switzerland, you can see here, do I see this sequence anywhere around the world? And you can quickly search and then pull it up and say, aha, I'm now seeing any evidence of this sequence or anything like it. And also then visualize some of these graphs. You can actually play with the sequence data and move them around should you so desire. So, building out this infrastructure or something that we're now, getting out so other people you can have this planetary scale index. So anytime we see any piece of DNA that's ever been observed, we now know when and where it popped up and it's similar to anything else. So if there's any sequence we find on those samples that come back from Mars, we'll immediately know whether or not it was related to that mission or a different mission and quickly prioritize it. Okay, so this is really big mapping and measuring efforts, but I want to switch gears in sort of the next phase of let's stop just measuring things and begin thinking about being more active with our defenses. So essentially, a few decades from now, I imagine we'll begin long-term human trials on genome engineering, particularly in some cases human, which I'll show you some examples of. And because you would have to basically have a multi-generational clinical trial to ensure that any armor that we embed into humans doesn't cause more harm than benefit it might make. And so the reason we want to do this is because we know that astronauts can have damaged DNA that is being ejected during flight. You can see here essentially, these 59 astronauts, they had 8-oxoguanosine, this damaged DNA coming out of the urine during flight. And this was just going into the space station. So if you think about what Scott Kelly endured was this 146 millisieverts of radiation, Mars would be the hardest mission yet at 1200. It would be a lot more radiation. And it really raises the question of, can we genetically protect astronauts, right? And really, should we do such a thing? Is there an ethical argument as to why we should? And we know in some cases, the arguments for, say, editing humans or editing embryos have been done in a really awful capacity. So for example, this is the way you should never do it, is say, I've CRISPR'd babies and I've released them onto the world and here they are. So this is definitely what we're not doing. The thing we're doing instead is having a long discussion about this first and talking about safety mechanisms and regulatory processes. One of them is with the Genome Project Right Consortium, where we're aiming to build human genomes from scratch as well as other genomes. And, it seems strange, this idea of synthesizing, engineering, designing genomes from scratch, but it's actually already in use. So for example, there's something called CAR T therapies where we're engineering cells for cancer therapies that are already widely deployed in China and elsewhere. This is us visiting a clinical site here. And you can see here that CAR T cells are chimeric antigen receptor T cells. You basically make a modified T cell, so it actually has a new receptor, you grow it up and then reinfuse it back into your body. And basically then you have an engineered cell that you've modified the genome of, put it back into the patient for therapeutic purposes. And there's actually, you think, it's not just one or two of these, there's actually now hundreds of clinical trials. Dozens of them are just for the CAR T or variations of CAR therapies. And so these trials, mostly in the US and China, but they're rapidly expanding around the world as this way of engineering cells and getting them into patients. And so again, here, they're clinical trials. So they're done carefully, slowly, safely. But we know they're already being done therapeutically as we speak. And so if this is being done, what if we could do other modifications to human cells? For example, we could add extra copies of P53. This is a gene that elephants have 20 copies of the gene, whereas we only have 2 copies. If you look at other species, how many copies they have. And they have a lower risk of cancer if you take that gene and put it into human cells. So that's interesting. What about the tardigrades? We'll go back to the cute little water bears. They actually have an interesting protein called DSUP, a DNA damage suppressor protein, that actually can give a 40% reduction in DNA damage if you just temporarily give it to human cells. But we've been working in our laboratory to say, well, let's actually do it permanently in these cells and make it constitutively active, so it's always there. And you can see that it quickly moves to the double-strand breaks. You can see these little dots here is where DSUP is, and the breaks here from H2AX, you can see that's where the double-strand breaks are. So we know it's actually moving in to kind of keep the DNA safe and help actually recruit repair processes. And you can also see here, we can get down to about an 80% reduction in the DNA damage, which is dose dependent. So we actually know that it's possible to make, use evolutionary lessons from other creatures on Earth and apply that to human cells to make it so they can survive more radiation. So we and others have already shown this. But even if you think, okay, CRISPRing genomes or adding genes, it's still a little bit, you might be anxious about that. The other avenue we're approaching this through is just epigenetic CRISPR. So you think of CRISPR as a way to modify the canvas of life. You think about ways you can add A, C, G, or Ts. There's epigenetic CRISPR mechanisms where instead of adding or removing or modifying, you only turn things on and turn things off in a very targeted fashion, using some of the same Cas9 systems. That's the other thing we're doing in lab. And we're not the only ones. There are other groups, actually, including DARPA, to say, what if we could preemptively express protective alleles in response elements? Or what if you could turn on genes as you go into a hazardous area, for example, a high radiation area for in this case, a soldier or a biological threat, and then turn them back off later. So you can have a transient and controllable ability to activate genes when and where you need them. And so these two areas of both genetic CRISPR and epigenetic CRISPR give us an extraordinary toolbox, only as of today, but I think it'll be even better in 20 and 30 years from now, that let us control exquisitely not just human genes, but potentially other genes that are brought into human genomes, and also would increase their safety potentially. Okay, so we're getting out a few decades from now. I think by the year 2050, we could get to the point where we might even have what's called Mars Base Camp. This is a rendering of what should be an orbiting space station around Mars by the year 2050, is a rendering from Lockheed Martin. And so by then we could start to think about, like, if we have protected human genomes or more enabled human genomes, what could that look like? And could they do well in spaceflight environments, or at the very least, the cells we could be testing at that point? And so on my wish list of things I would like to change is, one really interesting thing is, we have, if you look across evolutionary history, vitamin C, the ability to make vitamin C by yourself, was actually lost among some mammals, especially primates. You can see there's a mutation in our gene for this called LULO. So most of the dry-nosed primates down here especially don't have it, whereas some of the wet-nosed primates, a few of them still kept it. And for example, your cat and your dog can make their own vitamin C if you're looking at a cat or a dog right now, whereas we cannot. We have to get it from our diet, or else we get scurvy. But what's interesting is that other mammals still have this biochemical pathway, and we still have the pseudogene. We have a little bit of it left in our genome. We just need to turn it back on and tweak it. So why not bring it back? That's one thing we could do to basically begin the end of molecular ineptitude. Could we just put things back the way they were before and maybe give ourselves an ability to make vitamin C? But while we're at it, we have actually 9 essential amino acids. Why? Why do we have these in our diet when we know that there are other organisms on this planet that actually make all these same amino acids? And you can see the human biochemical pathway here is in black, but other organisms that we know can have enzymes that can create these essential amino acids already exist. It's just about repropagating them and putting them into human cells stably and safely and slowly. But this is another example. If we could be what's called a prototrophic human, where we could make all of our own amino acids and vitamins, it would make long-term spaceflight much easier, so we wouldn't have to depend so much on food supply. We could just maybe have Hershey's bars or other candies potentially. And so is in that regard, but then eventually in the 2050s, I think we'll get better and better at repairing disease genes. So molecular ineptitude refers not just to, creating vitamins and proteins, that's all wonderful, but what if we could actually avoid diseases in the first place that are really severe? So for example, this has already been shown in embryos where researchers such as Shirkat Metalbapov have repaired genes that create heart defects and just fix them before essentially the embryos develop any further. So you can fix it from the very first cell. And this idea of every cell being used as a way you could repair things or fix them before it's too late, it opens up other new doors where every cell has some degree of kind of liberty, where, you could essentially imagine if any cell can be used for any other cell therapeutically, any cell could also be used for reproduction. So we know that in mice, for example, there's already been examples of 2 mom and 2 dad children being born. So you can actually do slight modifications to enable reproduction from 2 males or 2 females. It's already been done, being talked about for human trials. And in human trials, it's already been done that there are 3 parent children, where you get a mother and a father, and you also then get a mitochondrial donor to avoid specific mitochondrial diseases. So this idea of multiple parents contributing to a child has already happened. And it really creates this question of, could you also then have other exowombs or artificial wombs carry them to term? That also has rapidly developed over time. And it really kind of creates this new paradigm of total cellular liberty. From the very first cell, that you are no longer beholden to the lottery of genetics that you've inherited. you have liberty to have things be modified or improved. And so, which opens up an idea of liberty to cellular liberty, but it also, of course, raises really stark and large ethical questions. In particular, can we really select embryos well at all? Do we have any sense of doing this today? There's a really interesting post about this from Lear Patcher recently that talked about there's a company called Orchid that's offering embryo selection for couples. And, he critiqued this as many others have in the field that say, well, how well can you do this? And how can you take an embryo and say, I'm going to perfectly predict which combination of genes you should have? Like, how good are we at this today? The way it works today is something called the polygenic risk score, which, as you can see here, this looks like a complicated equation, but it's pretty straightforward. It's a sum of, you just take all the genetic markers we know about and their different loci, and then you weight them to see, well, how good can we predict a score? So, for example, do you have a high risk of a polygenic risk score or low risk? And when will this disease potentially occur in your life? And you obviously want to get things that are lower risk. You want to lower your risk for any disease. It sounds simple enough, except the problem is it's been critiqued by many people in the field. So imperfect measures make imperfect metrics. And for example, if depending on the ancestry of a person, sometimes if you say you want to get a risk for schizophrenia, that actually risk is more dependent on your ancestry than it is actually on your true risk of schizophrenia, just because our ability to make these predictions today is very nascent, is very new, but it's not zero. It's just a question of how well can we improve it and correct for some of these factors and get more and more data. Actually, one of the best examples is the UK Biobank, which is linking whole genome sequencing and clinical metadata with all their molecular data, so we can make much better predictions at a large scale. And I think in the future, if we're looking out 30 and 40 years from now, it's estimated that what I propose in the book is that we should have a much more comprehensive and even a multi-generational risk score. So say you want to look at a specific risk score for a disease or a given treatment in a given place, even on a different planet, is what does this do to your life expectancy? What's the odds of the treatment being successful? What about your quality of life or those in your family or your community? You want to look at not just what it does for you, but what does it do for everyone else in terms of the economic of a burden, or is their severity increasing across generations, or does this one gene have multiple functions? And what does this do to the function of the onset across society? And so I think having a more inclusive, just molecular as well as social economic integration of some of these questions, in theory, should lead towards a more just and actually accurate deployment of them over time. And so my hope is that in 30 or 40 years from now, we'd start be doing something more like this versus just a simple, let's sum across all of your risk scores and put them out there. Okay, so we're getting exciting, or I'm getting excited. So we're getting out further and further into the future. We're now getting out towards the end of the century. And I think at this point, we might be able to have, some of the missions that are trying to get to Europa or other planets on the edge of the solar system, we might be able to synthesize genomes and begin to hopefully start to get people going to other planets. So the hope is we could do what we're doing here, but do it faster. We can already synthesize entirely new genomes, basically, and modify the structure of them. But what if we could synthesize other things? Like, wouldn't you perhaps want a chloroplast? Like, would you like to have green skin so that you could photosynthesize just by yourself? And so, if you're just like saying, I'm laying by the pool and I'd like to go lay out my skin because I'm hungry, you might be wondering, well, how much skin would I need? Well, we've done the math on that. So it turns out if you had green skin, you laid outside for about an hour, assuming a certain amount of your skin exposed, like half of your skin's exposed because you're laying on your stomach, a certain amount of loss of photons or photon capture, and over time, you'd probably need about a 300-fold epidermis expansion, which is about 2 tennis courts. So if you just laid out all your skin for about an hour, you get all the energy you need for the rest of the day, which might be kind of a gross image to think about mentally, but I think it's kind of fascinating from an energy perspective. But just means, it is potentially possible in some future world that you could do this. And, here too, it seems kind of farcical to say, could you have chloroplasts in your skin and be like a plant? Except that it isn't. There's already instances of human pig chimera embryos and multiple movements of cells between species, and even human organs being recently grown inside pigs. So things that are equally peculiar on the weirdness scale, but are already occurring today. So again, everything I propose in the book is only based on things that we know that are possible today or that are already happening today. It's just a question of degree and of accuracy. And so, okay, so maybe by the end of the century, some of those experiments are really starting to come to a head. And I think then in the 6th phase, we start to learn even more about the extremophiles on Earth, to use those as a substrate to have all those evolutionary lessons of survival begin to be deployed for human cells. So in particular, we have a project in the laboratory, which I talk about in the book as the Extreme Microbiome Project, looking at really peculiar places around the world where organisms have somehow learned to adapt and they've survived. And so again, we can use all these lessons of life from all the strangest places on Earth and someday other planets to help inform what can we do next to enable humans to survive in those places, or even get, even if we don't integrate those genetic elements into our genome, we can at least take their proteins and use them as therapeutics. So, for example, one of the things we found in the ISS microbes I talked about that could possibly survive on Mars, they had new genes and new proteins for DNA repair and biofilms and even sunscreen. Maybe we could use some of these organisms to figure out a way we could survive. If we're going closer towards the sun, we'd certainly need better sunscreen, or at least better molecular tools to defend against that radiation. Okay, so looking even further in the future, we're starting to get out to the next few centuries. And I think eventually we'd get to a phase where we begin to plan to send new genomes to seed Earth-like planets. And so this would be a violation of planetary protection, but this is, in my opinion, an inevitable violation, which I'll get to at the end of my talk in 5 or 10 minutes. Is the, if we start to send organisms that could survive there, we could imagine that we begin to actually prepare Mars and other planets to be self-reliant. So instead of always having to ship things there, we could eventually see this pie graph as it's mostly self-reliant, where you have, on-demand pharmaceuticals and therapies, large-scale fermentation and crop development on Mars, and you have integration into specialty materials. So we've published a roadmap of how we could actually get Mars ready actually for human like basically settlement, and also for self-reliance eventually. And akin to what we see for cellular liberty, this would be the beginning of planetary liberty, where actually the cells that you have can be created such that you can survive on one planet or another as much as possible. So it is kind of the ultimate in terms of liberty. The liberty is to have the most choice possible. And if we design ourselves correctly, you would not be essentially marooned so you could only live on Mars. You make it such that you could adapt and change it so you could live on Earth or Mars or an asteroid with very low gravity. And this means we'll have to start to look at all kinds of other planets and get a sense of how similar are they to Earth? What is the Earth similarity index? You can see here is 1, because Earth is where we're at now. We're trying to get as close to 1 as possible. And how many planets have we found that are within a certain distance that might be close to Earth in terms of our ability to survive on them? So we measure this. There's multiple formulas proposed in literature. This is measured by the density of the planet, the sort of solar flux, the equilibrium temperature of the surface, and other features of the planet. And the more of these bars that are filled in, the closer it is to being like Earth. And so if you've got a 1, you're very much like Earth. If it's above 9, it's blue. If it's purple, it's closest to Earth, but not that close, but white and gray, or we don't have enough data, or it's not that close to Earth. But you can see even within a few parsecs, or really a few dozens of light years, we have a few pretty good candidates that are not that far away, that are kind of close. If you look a little bit farther away from Earth, you can see here there's some other candidates that are a few more parsecs away. But we have some good candidates that have already been identified. So if you look actually at what's going to happen, that's what happened now. If you look at what would happen over the next few hundred years, this is just what happened really since 1992 was the first exoplanet. We had at one point 0 exoplanets that we could think about going to visit or to land on. Now we have thousands of them. Several hundred of them look like this, where they are planets that we could potentially land on, and they would be habitable, meaning they might have liquid water. They would be at a decent enough gravity that we could probably survive when we get there. And again, going back to this Earth similarity index, we can actually see these are kind of reasonably sized planets. And again, go back to these colors. If you look out within their sort of near galactic sort of neighborhood in 20 parsecs, in these blue spots are all these places that are pretty close to Earth, that if we got to, we could survive maybe even today, even without much modification or engineering or therapeutics. We might be able to make it. So in some cases we don't have enough data, but you can see in both cases we have areas that we could likely get to. And so I think what's exciting is this is just within 20 parsecs. If you look out 5,000 parsecs out to where we are within our galaxy, these are all the exoplanets that have been found. And you can see here this band right here. If you look at it from different angles, you can see here, this is because this is where we've pointed most of our telescopes. We've only really been looking for a few years at high dimensionality and high density, and we haven't even really explored those other places. But we already have all these blue planets that are not that far away that we could potentially reach and potentially survive at. So really extraordinary sort of opportunities within a pretty near area of our sort of galaxy. And so to do that, though, the final phase, or ninth phase really of the plan, the penultimate phase, is that we think about, okay, currently I'm presuming no new methods in propulsion that would get us there faster. So if that's the case, maybe I'll be proven wrong. I would love it if I was. But if we assume there's nothing magical that occurs, we basically need probably 20 generations of people living and dying on the same spacecraft to make it to near one of the best exoplanets. And so what that means is you have an interesting ethical and technical challenge where we say we want people to live and die for 20 generations on the same big spacecraft. And this too is kind of weird. You think, well, how could that be possible? Well, people get really bored. Yes, they'll probably will get bored. People get bored on Earth though. And how could you actually keep them safe and sane? And I think 20 years ago, it was hard to imagine. But today, we've all recently lived through a pandemic, so we all gotten pretty good at staying in one spot for a long time. And not that most people liked it, but we've learned the benefit of being able to access the totality of human culture online, like through Netflix or movies and entertainment, culture, music, all these things at our fingertips gives you a lot to do. And then also we would need to have these people be viewed as sort of the real pioneers of humanity so that we could actually survive in a distant solar system. And so, which I think is possible now today because we have entertainment options. And the second thing is we know a lot more about biology than we ever did before. So we actually could keep them safe long term and have the technology to get them there and hopefully sustain themselves. And so my hope is that, toward this penultimate phase, we would have gone through this entire phase where actually have all these different technological developments in evolution and epigenetic technologies and genome engineering and transplant medicine, developmental biology, and all the sociological implications, that when we get out towards the last few hundred years of this plan, by then we're seeing actually programmed planetary protection. We're seeing adaptable traits in Mars that have been identified, and we have prototypes for generation ships that we begin to ship out by the year 2500. Eventually, we have engineered organelles. We have even occupational enhancements for surviving if you're working on a harsh planet, or eventually these become recreational enhancements. And it's possible in the future that babies might be genetically corrected or selected just to ensure that they, assuming we get the polygenic risk scores right. I'm thinking that won't be for another couple hundred years because it's going to be hard to do, but eventually, hopefully, it would. And you could have mentioned autotrophic human cells that can make all their own amino acids and vitamins and survive on another planet just fine. And if at some point the first deep space baby might be born, the first native Martian, or a couple that was born on Mars had a child on Mars, and ideally life expectancy would also get better. So this is, the hope for the next real 500 years, that at the end of that time frame, we'd send a generation ship to get people out towards a new star. And there's often a big question that comes up as, well, why do all this? Like, what we have other problems on Earth. We have poverty, we have social injustice, we have many systemic problems, including racism and other just challenges in society. Why do this? Why not focus on those? And to that I say, we can do two things at the same time. We can walk and chew gum. We can, as we have done before, we can land people on the moon and still build an economy, as well as progress in many issues of justice and equality and problems in society. And the other reason people often give for why we should explore is they just say, well, we are just explorers. Humans have always explored. That's why we should go, which I think is an okay answer, but it's not as good as the fact is that, we essentially are the only ones that are aware of this. So, for example, in the last few decades, about 1 in every 4 birds in North America is gone. It's just they've been lost because of habitat loss, mostly from humans, but from other reasons as well. And this is not the first time. If you look over the past 500 million years, there's been at least 5 times where anywhere from 75 to 96% of all species on Earth were obliterated and gone. And this is true in worlds with and without polar ice caps. And so what that means is, we want to avoid here is a sixth extinction, maybe the final extinction. But what's interesting is we are the only ones that knows that this chart is happening. We're the only ones that can prevent extinction. We can also cause extinction. We have a pretty bad track record on that so far, but we're the only species that understands this. And so if you think about, in the history of the Earth, if you put this in the context, over the past 5 billion years and what's ahead for the next 5 billion years, we are currently in a very nice but temporary temperature canyon where we have liquid water on the planet. And as I was finishing the book and writing it up, I'd always thought we had about 5 billion years left before the sun would engulf the inner planets, and that would be the end of the Earth. But it's actually about 1 billion years before the oceans will probably start to boil. And I actually got really upset when I was finishing the book because I thought, I just always thought we had 5 billion years. And I went downstairs and told my wife, Joan, and she said, well, why are you so upset? I said, well, I just thought we had more time. And she said, well, you still have 1 billion years. I said, yeah, but it's like I was thinking I was going to live to be 100 years old, and you tell me you're going to be dead at 20. It was kind of like a punch to the gut, because I really think this will, everything we've ever done and built and learned, even if we have perfect world peace, will all go away if we're still here. And so I just really thought we had more time. And I thought we have this, we're the only ones that know this is the case. So for example, when you were a child, you probably learned about the food chain, and you learned about there's 3 kinds of organisms, right? There's producers, there's different kinds of consumers, and there's decomposers. This is basically what we all learned as elementary school children. But what I'm telling you today is the reason we have to go to the other planets is because our time is limited, and we are the 4th kind of organism that exists that's new and nowhere else has it, as far as we can serve as the guardians, and we alone are aware of extinction. Only humans are extinction aware. And I think in the future, it's possible that machines could become extinction aware, and maybe they too could become guardians. I'm fine with that as well. But we basically have we're the only ones that can actually prevent extinction. For example, this is a black-footed ferret that was cloned from cells that were stored after 30 years to bring it back from the edge of extinction. We can even bring the woolly mammoth back from all the way from being extinct. And that's another project that's happening at Revive and Restore. And I think we have a duty to all life, past, present, and future, to serve as the guardians, to serve as the shepherds for these life forms. Because if we don't do it, as far as we know, no one else will. And I think the moral argument is that whatever your ethics might be, whether you're utilitarian or if you have Kantian ethics or if you're Rawlsian, all ethics depend on existence for them to be applied at all. And so existence it precedes the essence of anything, including any ethical framework. And so the primary and fundamental first ethic is simply just to survive, which we have to do in the long term. And if we do it long enough, though, we reach the final phase, which is the big question of, if we should prevent the implosion or entropy death of the universe, or should we let the universe end in the hopes that life might begin again? And I think we would survive. I think we would try and preserve life for as long as possible, because there's no guarantee that life, which is extremely unique in the universe, as far as we know, would ever arise again. So in that regard, I think we would even try and restructure the space-time of the universe itself if we could, just to survive long enough after long enough time period. And I think it's actually a unique duty that we and we alone can understand and act upon. So it really gives a duty for all of humanity, which I'm hoping that we enact and also do together. So with that as a closing, I want to thank a lot of the members of the Twins PI teams, which really inspired and led a lot of this work. And it was really a pleasure to work as these 10 different teams. Thanks to Mark and Scott Kelly and other astronauts, who, as well as Kate Rubins, who've given quite literally blood, sweat, and tears to these missions. And also thanks to the lab, who's inspiring and makes all this possible, and also is taking recently fantastic pictures. Thanks, of course, to funding from NASA and the NIH and World Quant and the Gates Foundation. Thanks to many friends, collaborators, and colleagues. And thank you very much to the Royal Institute. And thank you so much for your time.